Okay, that's great. Thanks. And thank you very much for this opportunity. I really appreciate it. I've been told I've got 20 minutes. I'll try and stick to that. Yeah, I guess to echo some of the things that have already been said about the in-person meeting in Birmingham, I enjoyed that thoroughly. So I hope I'm looking forward to the next one in Edinburgh. And thank you for the organising group for making all of these things very easy for us. Yeah, so I'm currently based at Northumbria University. I guess I'll just say a little bit about myself. I'm at the moment, I've got this job as I'm on a 3-year fellowship, which converts to a full professorship at the end of 3 years. And one thing I would like to say is, I love collaborating. So I just pulled this from my pure profile, and I've collaborated quite widely over the years. I'm also old, so that helps in terms of those collaborations. But I've also worked very closely with industry, working with some startups and Fortune 500 companies, all of which have their different kind of benefits and challenges, and also, of course, a lot of academic collaborations. And just to say a little bit about myself, I was at Northumbria University in the 90s up till 2007, where I then moved down to Melbourne, Australia, or as director of Centre for Human Psychopharmacology at Swinburne University in Melbourne. And then actually, before I returned last year back to Northumbria, my old department, in fact, I did spend 5 years as a chief scientific officer of a small New Zealand company called Arepa, trying to make evidence-based brain health drinks. And I'm actually going to touch on that a little bit in this talk. So that's my background, and please contact me if you're interested in collaborating or talking about any of this. In terms of my research, I guess can you see my cursor, by the way?
Yes, we can.
Okay, so most of the work I've done has been, I guess, looking at randomized controlled trials, looking at these, I just call nutrient interventions, so diet, nutrients, botanical interventions, enriched foods, and as well as just trying to capture the effects to understand the mechanisms of enhancement. And also over the years and recently, I've also dabbled in drugs and looked at the effects of alcohol, and particularly alcohol hangover and the effects of aging. So those are my main research areas that are much more focused on cognitive enhancement and neuroprotection. And I'm just very briefly going to talk about alcohol hangover, because it's probably one of the few areas of my research that really does speak to at least the G and I of GABA, but particularly the I. So this is a kind of summary slide of what happens with alcohol hangover, and there's this intoxication phase followed by a hangover phase. And what you can see is that behaviorally, the two things are quite distinct. So although performance suffers with both intoxication and hangover, subjective performance, people often think that their subjective performance is better when they're intoxicated, which is a huge problem for things like driving. And also, of course, there's increased mood in the early phases, whereas hangover absolutely flattens everything. And our best evidence to date is that this is due to a cytokine response to ethanol itself and to some ethanol metabolites. And the reason that I'm including this is that this is a really under-researched area. So if we look at alcohol intoxication, there's something like, I think, 80,000 publications, whereas for alcohol hangover there are about 200. So this is an area that's really under researched, but it's a very important area, I think, given the kind of functional consequences. So if anybody's interested in collaborating in that field, please let me know. Okay, I'm going to leave that there and talk about, I guess, the main areas. So people will be very familiar with this kind of slide. The idea that this is actually Alzheimer patient 1, Auguste, who was diagnosed by Alzheimer in about 1905-06. And if she was around today, of course, she'd be given one of the cholinesterase inhibitors, or maybe memantine, or more recently, one of the monoclonal antibodies. And these treatments have been spectacularly unsuccessful in treating Alzheimer's disease. So there's a growing consensus that preventative interventions are best initiated probably decades before the clinical manifestation of Alzheimer's disease. And when we look at the sort of interventions, this is the Lancet Mission version 2.0, published a couple of years ago, showing that although it doesn't explicitly mention diet, there are a number of diet related processes like cholesterol, diabetes, hypertension, etc., obesity, excessive alcohol, which could reduce the prevalence of Alzheimer's disease significantly. And when we look at the kind of dietary interventions, they include things like flavonoids, the Mediterranean diet, which I'm going to talk about later. And I'm just going to give one example that we researched from my own study, which is curcumin. And I present this slide a lot because it's quite good fun. I have to apologize because my PhD student at the time who made this slide had just discovered PowerPoint animations. But here we go. This is curcumin. And we'll just wait for this to play out. This was Kate Cox who made this slide, and she loves curcumin, as you can probably tell. But it does illustrate an important point, which is that a lot of even the individual nutrients, bioactives that we isolate, have these pleiotropic or polypharmacological effects. And in this case, the ones with this sort of rosy glow around them are relevant to cognitive aging and cognitive impairment. And so for a number of reasons, after a series of studies, we decided to focus on hippocampal function. And you'll be aware that of the brain areas that are studied and show age-related changes, most show a fairly linear change with aging. The hippocampus is slightly different. It shows this little kink as we hit about 50 or 60. So there's this slightly non-linear decline. But the other thing that's interesting about the hippocampus is that it is exquisitely sensitive to lifestyle interventions, particularly exercise, but also dietary interventions. And it is one of the few, maybe two areas of the brain which are capable of adult neurogenesis. And so this is I guess I just want to use this as an opportunity to talk about some of the neuroimaging techniques that I've been using over the years. One of the reasons that I moved to Melbourne was they have fantastic neuroimaging facilities, both fMRI, but also MEG, which picks up changes in the magnetic field of the brain. And using this methodology, you can essentially insert a virtual electrode quite deep into the brain, including the hippocampus, and using very sophisticated deconvolution algorithms, you can measure activity from these deeper regions. So this is an example here of what we used to test hippocampal version. We used a virtual version of the classic Morris water maze. So this is the kind of classic Morris water maze where you drop a rat or a mouse into cloudy water, and it has to navigate to find a hidden platform, and gets much better at finding it using cues, usually in the environment. And this is the human version where we don't drop the humans into cloudy water, but we put them into this virtual environment where you can see that the pattern of finding the virtual hidden platform is very similar. And using MEG, we can pick up hippocampal activation, which shows a very similar pattern to the pattern that's shown in animal models. So this is a very nice way of measuring hippocampal function. And using a study where we supplemented with curcumin, a very highly bioavailable curcumin, for 12 weeks, we found a significant improvement in performance on this virtual water maze. The other hippocampal task that we used in the same study was pattern separation or mnemonic similarity. So if you're not familiar with this task, what happens is you're presented with a series of objects and you have to say whether or not you've seen it before. And some of the objects are identical, like these pineapples, but some are very similar but slightly different, like these Rubik's cubes here. And so you can make the lures, the new objects, very similar or very different. And basically, depending on how similar they are, the more similar non-identical stimuli presented later really work the hippocampus. So a lot of people will be familiar with this idea. And so these are some of our data showing hippocampal activation, showing these very similar lures. And this is what happens with age when people are presented with these sort of tasks. So this is nearly 100,000 people. And if you can't read this, it goes from about mid-20s to early 80s. So this age curve here is specifically the ability to correctly reject new stimuli. I hope that makes sense. And if we look at the effects of curcumin, as we move to the more similar objects here, you can see that there is a significant improvement. So again, this is good evidence of improved hippocampal function. And again, just to put this into some sort of perspective, if we superimpose this onto the age curve of our participants, you can see that this is a kind of, if you like, a cognitive rejuvenation of 5 to 10 years. Okay, so I want to sort of pivot a little bit to a slightly different aspect of this, which is looking at something which is quite interesting about clinical trials, I think, which is that myself and many colleagues over the years have suspected that the participants that come into our clinical trials aren't representative of the wider community. And the idea is that maybe we've got this idea where we've got these optimal nutrient status individuals, and we're thinking that maybe we can improve function in suboptimal and obviously these are clinical conditions, which I'm not going to talk about now. It's mainly this sort of suboptimal, what's sometimes called nutrient insufficiency rather than frank deficiency. So what happens here? And the idea has always been that, well, if we bring people into the lab who are sort of insufficient, that maybe we've got a better chance of improving their cognition, better chance of getting a signal. But are the people who we bring into the clinical trials actually representative? And so in order to do this, we recruited participants. This is just for a multivitamin supplementation study where we tried to find people who were suboptimal in terms of their diet using a very simple diet screen here. And as we suspected, in order to get the suboptimal group, we had to screen far more people, about 4 times as many actually. So in our initial screen, well, to put it into perspective, about 5 to 10% of Australians meet the criterion of fruit and vegetable intake. In our initial screen, 75% of our participants did. So there's a 10-fold disparity between our volunteers. So that kind of validated our idea that the people who are coming into our lab in the well-to-do eastern suburbs of Melbourne were not representative. But when we managed to get a group that we screened far more people, where we found enough to match for the optimal diet with suboptimal, my prediction was very clear that the suboptimal, the people with the suboptimal diet would differentially benefit from supplementation. And what we found was absolutely the opposite. So this was really interesting. So what we found was on our primary outcome, which was pre-registered, was performance on the Stroop test of selective attention response inhibition, we found that the people with the optimal diet were the ones that significantly benefited, whereas those with the suboptimal didn't. So was completely wrong, but it actually raised an issue which I think is really important for our studies, which is that it may be that some people, based on their initial nutritional status, may not benefit from our sort of interventions. And I'm going to come back to that. I should also say that this is not the case always, so we have to be very careful here. So people here may be aware of the Cosmos trial, a massive trial looking at using a factorial design to look at cocoa flavonols and a multivitamin intervention. And sorry, these are very messy slides from the papers, but what you're looking at here is what you need to do is compare the dotted lines with the filled lines of the same color. And what this showed, particularly for cocoa flavonols in this case, was that the improvement was seen in the people with the lowest baseline flavanol intake. So this was kind of the opposite to what we'd shown in the multivitamin trial. But I think it just shows that this is, it's really important to consider the baseline characteristics of people in clinical trials. And I'm going to talk very briefly now about something completely different, which is about some of the work I was doing as Chief Scientific Officer, where essentially this was a really interesting kind of discovery, I thought, where I was very peripherally involved in this, but just to go through it. So this is some early work from about 2015, showing that in human feeding studies, when humans are fed this particular New Zealand blackcurrant containing different levels of anthocyanins, nearly 600 mg compared to about 80 mg in a powder compared to a juice, this shows almost complete inhibition of monoamine oxidase. So this is a monoamine oxidase inhibitor in human feeding studies, and these are the equivalent of a couple of handfuls of the blackcurrants themselves. I don't think this has been demonstrated from any other food in human feeding studies, although there's a lot of in vitro and animal evidence for the effects from other berries, for example. And this led to a attempt to try and discover what the ingredient or what the component of the blackcurrants might be. And essentially what this found was the discovery of a new molecule a new molecule in the context of brain health called sarmentosine. So sarmentosine is a glycoside which was discovered through this really very hard work kind of fractionation and refractionation of different fractions of the berry. And what this found was essentially it discovered this molecule sarmentosine, which shows this monoamine oxidase inhibition at a level which is not much weaker, but not completely dissimilar to some of the pharmaceutical monoamine oxidase inhibitors. And this then led to a first into human trial of sarmentosine to see if it did indeed, just to verify that this was responsible for the monoamine oxidase inhibition. And what you can see with 2 doses of sarmentosine here is, again, almost complete monoamine oxidase inhibition in this case. And I'm really just putting this up here because I was messaging the scientist who was actually looking at the results as they came for this final study, as they came off the machine, probably on a screen actually rather than the machine. And it was quite interesting that somebody who's been conducting these sort of trials for many years, the idea of discovering a new molecule in the context of brain health wasn't something that I thought I would do as a as a chief scientific officer of a New Zealand startup. I thought that would be something I might be doing more in an academic context. So it just goes to show. But I would say that, you know, we're trying to take this further. So if anyone's, I know some of the people on this call might be involved in some of these studies, including the group at Leeds Beckett. And so I think it's also just worth saying that the relationship between monoamines, oxidase inhibition, and mental state in these studies is, you know, it seems to have some real meaning there, in that it correlates with a number of positively correlates with these positive mood states, proficient, strong, quick-witted attention, well-coordinated, energetic and tranquil, and negatively correlates with mental fatigue. So this might be something that will be quite interesting to take forward. And I'm just going to finish off this first part of my talk now, just to say this is some of the people in my lab over the years I've collaborated with, who are largely responsible for the work I've presented. Now, if you can turn off the recording, I will just spend a couple of minutes talking about the final thing, if that's okay.